Today, the topic of discussion is polymerase chain reaction, abbreviation is PCR. The PCR is a technology, it is prepared by Muhammad Ijaz, MPhil Zoology. In this lecture, the PCR outlines are first one is the introduction, principle, components, procedure, advantages, disadvantages, and last is the applications. First one is the introduction. You know that a human genome, after the human genome project, the scientist said that it is made up of 3 billion chemical base pair, in the form of A, C, T, G, and C. Scientists often, they need tiny segments of DNA because, as we know, DNA is very, very large material. So a teeny segment sometime have to need that it is made many, many copies so that it could be studied. So there is the PCR technology which came into the point, any piece of DNA that could be quickly amplified or in copied many many times purely in the lab. As far as discovery of PCR is concerned, a scientist which is named by Carey Mullis, on a long motorcycle drive, he is thinking, mentally visualizing the process, and it is also got the Nobel Prize in 1993. As far as key principles of PCR are concerned, there are many rounds of DNA synthesis. DNA synthesis actually required certain substances, or you can say that raw material for this process. It is it is template, usually DNA, and primer. Primary, it is a short, tiny segment of DNA which is used for the PCR. Third one is the polymerase, which is an enzyme which can actually amplify the DNA piece. And last one are the nucleotides in the form of raw materials. The no, primers are the target DNA for amplification and face inwards. DNA synthesis therefore proceed across the region between the primers. Each round of DNA is called cycle. Cycle means the process where it is start and after some time it reaches at that point after some time. So that is called the cycle. And this is three reaction steps. One is called denaturation, second is called annealing, and third one is called extension. The products of each synthesis reaction can be used as template for further DNA synthesis. This reaction is called exponential amplification, means the previous DNA material could be doubled after every cycle. So there is the a graphical or imagination approach to explain the PCR procedure. Logarithmic application. There are two DNA segments. After first round of cycle, they could be 4, 8, 16, 32, 6, and so many of them. Uh, PCR can generate 100 billion identical copies of a specific DNA in a matter of hours. It depends upon the situation that what technique you are using, generally it is normally 1.30 hours to 3 hours. It will take time to this procedure, and it's all done in the test tube, reagents and heat. Components and reagents as well as DNA template, primers, Taq polymerase, DNTPs, buffer solution, divalent cations in the form of magnesium ions or manganese ions, and monovalent cations such as potassium ions. DNA template, you know that it is a picture of DNA in which we can see that it is the DNA backbone. This is, phosphate group, and this one I should change the pointer color, and this is the sugar component. ठीक है, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. This is the sugar phosphate backbone, which is the main part of the DNA. And this DNA template, you can see that phosphate group, that is also called, there is the 5 prime, and because if we numbering the sugar molecule, we can see that this carbon is carbon number 1, 2, 3, 4 and 5. This carbon number 5, which is being attached with the phosphate group, so this end is called 5 prime end group, and other end of the DNA molecule at the sugar end, it is the 3 uh, carbon which is attached with the OH, free OH group. That's why this end is called 3 prime end. Every DNA molecule should have a free 5 prime end and free 3 prime end. This is and we also in the DNA model we see that these are two strands which are anti-parallel. Anti-parallel means one end is 5 prime, one end is 5 prime, and other end is other second end, other 5 prime end below, and upper is the 3 prime end, and there is also 3 prime end. So if you want to see the certain or more detail of this DNA template, you can simply ask me any question in the comment section. Next are the primers. So you can see that primers are the small segment of DNA which can complementary to our DNA template. You can see that these are the primers, red one, these are primers, this is a primer, and this is also a primer. This one, okay. Okay. These are the primers which are complementary to our DNA template. If our DNA template has C, our primer should contain, here is the G and T, A complementary. Primers are short pieces of DNA that are made in the laboratory. We are synthetically synthesize the primer according to our gene of interest. And in PCR experiment, two primers are designed to match the DNA segment. Here you can see that one is this primer, and second one is this primer. Next is the temperature chosen for PCR experiment, determined by the primer sequence and the requirements of the DNA polymerase being used for the experiment. And denaturation temperature, which is almost around about 94 degree to 95 degrees centigrade. For selecting the correct hybridization temperature is very critical. If the temperature is not suitable or complementary to our DNA, primer will not bind primer will not bind to our DNA, you can see that annealing will not be occurring. So next is if the temperature is too high, primers will not bind to the target DNA. If temperature is lower, then also the primer will not bind to the segment DNA. Or second, if the temperature is very low, there are some non-specific segments of DNA causing amplification of non-target sequence. So hybridization division determined largely by the A plus T and G plus C composition of the primers. If primers contain high percentage of G plus C, then should we have high temperature for the primers to bind with the DNA template. Ideal temperature for the hybridations are calculated on the G plus C and A plus T percentage. Here, primers are also necessary because DNA polymerase cannot attach at just any old place and start copying away. It can only add on to an existing piece of DNA. So in this diagram you can see naturally occurring in the cell that this is the primer, this is a target strand, and this is the RNA primer which is complementary to our DNA template. This is also the primer, and when it is bind, RNA polymerase can synthesize the new uh, segment of the new template DNA in the 5 prime to 3 prime direction. Next, we can study the DNA polymerase. DNA polymerase is a naturally occurring enzyme. You know, enzymes are the complex protein. Its function is to copy the cell's DNA before it divides into two. Tag polymerase, it is a DNA polymerase that most often used in the PCR comes from the strain of bacteria called Thermus aquaticus. There is a bacterium. This is the picture of Yellowstone National Park. This is a picture of Yellowstone National Park where the this Thermus aquaticus heat stable bacterium could be found, and these bacterium naturally contain the enzyme that is called tag polymerase, or uh, in the which can used in the PCR. In 1989, the journal Science Journal called the tag polymerase the molecule of the year. Nucleotides are the building blocks of DNA. That molecules are made of. You add a mixture of four types of nucleotides to your PCR. You know that there are the four types of basically nucleotides in the form of purines and pyrimidines. These are adenine, these are guanine, these are cytosine, these are uracil, these are thiamine. In one, you can see, you also know that DNA and RNA two types of nucleic acids. In case of DNA, there is present A, G, C, and T, while in RNA it is present A, G, C, and U. Instead of T, it is there is present U. Buffer also required for PCR.